I will take one bit of poetic license here and say that one of my greatest accomplishments has been to recruit Obi Griffith. He's paid dividends in spades over the years, so congratulations to Obi on what he's done with his career. Now, my job today is to talk about a little project that we've been working on for the past few years, focused on the use of immunotherapy for the treatment of T-cell malignancies. And these are my disclosures. You know, CAR T-cell therapies, you've probably all heard about them. They've revolutionized the treatment of B-cell malignancies, specifically leukemia, myeloma, and large cell lymphomas. And, you know, we have not been able to develop such therapies for T-cell malignancies, which are actually more devastating for patients, both T-cell ALL and T-cell non-Hodgkin's lymphoma. And so there are lots of problems and lots of reasons why there have been no advances. And so we thought that maybe we could try to make a little dent in this field. The outcomes of patients with T-cell ALL, acute lymphoblastic leukemia, is very similar to B-cell ALL. And the survival rates, although our preconception is that adults with T-cell acute lymphoblastic lymphoma do better than those with B-cell ALL, it's not true, shown by these data from the MRC and ECOG, showing that the outcomes are about the same. But the most important thing is that if you relapse with T-cell ALL, you have only a 24% chance of survival at 1 year and only 11% at 5 years. So this represents a really devastating disease for which there are very little or no targeted therapies. I've also listed some of the genetic, since I'm speaking at a genomics conference, I thought I'd list a few of the genetic predictors of outcome, including things that you know about, such as KRAS and PTEN mutations associated with poor risk disease, JAK3 mutations, and of course, Notch family mutations associated with a good risk. You know, so these diseases, you know, there's a lot of them that are kind of uncommon as well. As pathologists and molecular geneticists, you've probably seen some of the data on these diseases. They're rare and they're devastating. They encompass T-cell and NK cell malignancies, and there are currently no targeted therapies for these diseases. This really ended recently with a beautiful paper in Nature Medicine by Martin Poulet's group at UC London, where he he realized that there were 2 genes that encoded the constant region of the beta subunit of the T-cell receptor, and he made an antibody to one of them. And making an antibody to one of them, you could actually potentially target half of all the T-cell malignancies, since it's a clonal population of cells. And you know, in your peripheral blood, your T cells utilize these 2 constant region genes interchangeably, and so 50% of your cells are going to be expressing one of the constant region genes and the other 50% the other. And so you'll have a mixture of these cells, but when you have a malignancy, they're all expressing one of the other. And so he made an antibody to one of them and then converted that into a CAR, and is now testing that in the T-cell malignancy field. And what's happened is that this is a great advance for mature T-cell lymphomas, but it doesn't apply to T-cell ALL because the T-cell receptor is not expressed on the surface of T-cell ALL. So you need to do something different. So we thought that maybe we could really pursue this by using CAR T-cell therapies, but CAR T-cell therapies are really limited in the treatment of T-cell ALL and T-cell malignancies because primarily the target antigen that is present on the T-cell malignancies is also shared on T-cells. And so that would mean that if you generated a CAR T to a T-cell antigen, you're going to kill it, you're going to kill yourself. It's called fratricide. And that has really been one of the major limitations. And also, in the peripheral blood, although they morphologically look different, these T-cell ALL cells, the phenotype of them is not sufficiently different from a normal T cell so that you can easily separate them. So you'll always have contaminating T cell ALL cells in any autologous product that you want to genetically manipulate. And so that would result in any genetic manipulation of that product would also be a genetic manipulation of not only normal T cells, the effector T cells that you would want to use for a CAR T, but also the malignant cells, which then potentially would be made resistant by your manipulation. And then finally, you could use T cells from an allogeneic donor, a different donor, but those T cells, if they get into a mismatched recipient, are going to cause life-threatening or life-ending graft-versus-host disease because that donor is not going to be matched. And even if the donor is matched, you can die of graft-versus-host disease. So we thought, and this was really work started in the lab by a really brilliant postdoc who's now the scientific director at a company that we started from this work, Matt Cooper. And we conjured up the thought that perhaps gene editing of CAR T cells to delete the target antigen that we were going after and the T cell receptor, which is the mechanism by which a T cell recognizes cells as non-self, would mitigate those two major challenges, and that is fratricide of T cells and graft-versus-host disease. And so what target did we go after? Well, there are several targets that are really pretty good targets in T cell malignancies, and especially T cell ALL, and that's CD7, as you can see here. This is a recent not so recent, a relatively old publication, I should say, showing the expression of various T cell markers on various T cell malignancies. And you can see that in T cell ALL, CD2 and CD7, especially CD7, is the best target. So we chose CD7 as a target, and it's expressed on 98% of all T cell ALLs. Interestingly enough, it's also expressed on 20 to 25% of AMLs, which is a really big population of patients. And so this potentially could be used for the treatment of AML as well. It's expressed on NK cells, and so it could potentially be used to treat all of these very rare and devastating NK cell malignancies, which you may have seen or have done genetic analyses of. And also, we had then proceeded thinking that CD7 would be a good target by knocking out CD7 in mice and looking at their T cells, and found that those T cells and their T cell subsets were normal and function normally. So we thought if we deleted CD7 in an effector T cell population, we wouldn't disable the T cells from functioning normally. We generated a chimeric antigen receptor. In this particular set of experiments, initially in the preclinical setting, it was a third-generation CAR T, and we made our own. We generated our own CD7 scFv, and we tested these in the context of both 4-1BB and CD28 co-stimulatory domains to allow the CAR to function. And we marked these genetically modified T cells by putting a CD34 epitope on them so we could purify them and track them in vivo by flow cytometry. We use CRISPR as a mechanism of gene editing for many reasons, and this was the product that we are taking forward in the clinic now. So this is what a genetically edited peripheral blood T cell looks like before and after editing for both the T cell receptor. So to get rid of the T cell receptor, we gene edited the T cell receptor alpha subunit. You need that subunit to actually bring the entire T cell receptor to the surface of the cell. If the T cell receptor is not expressed on the surface of a T cell, it can no longer recognize foreign cells as non-self. And so my T cells could be used to treat everybody in the audience. That's the beauty of this. Now, you could reject my T cells. I hope you wouldn't, but you could. And that is a problem that we're working on as well to try to overcome inherent host-mediated resistance. This approach, though, targeting CD7, because it's expressed on host immune cells, specifically NK cells and T cells, by targeting the host immune system, we would overcome, by definition, a lot of those issues with host-mediated resistance. So this is a double whammy, if you will. You can see that the cells in the left panel, they're positive for both CD3 and CD7, CD3 the T cell receptor and CD7. And when we do multiplex gene editing, we can get rid of most of both of those targets very quickly by CRISPR. And you can see that there's still about 4% TCR positive or CD3 positive cells in the right panel. In order to infuse those into a patient, we'll need to get rid of those residual CD3 positive T cells because those still could be enough to kill a recipient, and this is what the clinical product has done. So the first thing is to test it in mice, and so we used, in mice, we used T cells that were expressing a CAR to CD7. And we tested this in various settings. We used a cell line that was a CD7 expressing cell line. It was bioluminescently labeled. So the blue signal there is light that's being emitted from mice that have these tumors. So the more blue, the more, or red, the more tumor in these mice. And you can see that the mice that are treated in the far right panel with a CD7 deleted CAR T, you can see that those mice have essentially no tumor in them, and they cleared their tumors nicely. On the left panel on the bottom, you can see the red line looks like those mice on the bottom panel, the tumor is gone. That's the BLI signal. But on the top panel, still those, the red line does show that a bunch of those mice eventually die. And what are they dying of? They're dying of graft versus host disease. That's because the T cell receptor in this particular experiment was not eliminated, only the CD7. So they kill the mice from graft versus host disease, but they also kill a tumor. So we got to do both of those things. And so in this experiment, this is kind of a complicated experiment. I want you to believe what I'm going to tell you here. And what I'm going to tell you is that in the end, when you delete both the CD3, or the T cell receptor, by knocking out the T cell receptor alpha subunit and CD7, and then put a CAR into these cells directing the T cells to CD7, which is the target on the malignant cells, we eliminate all the malignant cells, and none of the mice develop any graft versus host disease, and they look completely normal without any pathology. So this, in our view, was the signal that we might have something that could work in human beings. We did do off-target analyses, and we used several techniques called guide-seq and oligonucleotide hybridization approaches to look at off-target effects, and we found that the off-target effects of our gene editing was very limited, and it satisfied the FDA and allowed us to move forward into the clinic. This technology was licensed, and the company that licensed this technology is now working on a clinical trial, first phase 1 clinical trial. And I think this is the tricky aspect of academic medicine, to be an entrepreneur, meaning to develop these technologies, and then to try to see that these technologies get developed by some of your own colleagues and not sending it far away. This has been very technically challenging, but we were able to actually open this study at Washington University, as well as a number of other centers around the world. And I have no role in this study at all, but the study is ongoing, and so our patients in the St. Louis region are going to benefit potentially from this treatment. Armin Gobadi, a colleague of mine, is doing the clinical oversight of the trial, and we're able to do the correlative science for the study. This is a simple phase 1 design where the patients get increasing doses of these. They're called universal CAR Ts. Universal means the TCR has been deleted and the target has been deleted and a CAR has been placed into them. So when universal means that, again, my T cells or your T cells could be used to treat anybody in the audience. And so you can imagine that instead of one product treating one patient, which is a normal CAR T cell product costing $1 million, this potentially, there's enough cells that can be isolated from me that could treat 100 patients in the audience. So it's potentially a different ball game altogether, overcoming many of the economic obstacles associated with CAR T cell therapy. So this is just a simple phase 1 design. We're in the middle of this now, and there's an extension phase to this. And this, if I had a pointer here, I could show you a little bit. I don't know if anybody has a pointer, but if you look at the left upper panel, you can see that the PET scan of that patient there's a lot of disease in that patient. After treatment, this is the phase 2 dose. Essentially, almost all the disease is gone. And if you look at the middle panel on the upper part, you can see there's on the lower, at about 8 o'clock and 7 o'clock, there's an orange group of cells. Those are CAR T cells. And there's a blue group of cells. Those are CAR T cells. And you can see on the bottom left panel at day 15, in this particular patient that had lots of disease, those CAR T cells are currently appearing. And those CAR T, you got one. This is fantastic. Okay. Well, that's terrible. Okay. That's good. All right. So yes, yeah, okay. So you'll have to those two blobs of cells that are the CAR T cells suddenly appear at the time that the disease is disappearing. So this is a proof of principle. This is working. This patient went into a complete remission, relapsed 6 months after treatment. But this was the first sign that this is working and it can get people into remission. So this has not been done before. So this suggested to us that this was an appropriate approach to treat these patients. This is a patient treated at the dose 4 level. This is the PET scan before and after treatment. And you can see that all that bright light in the bottom of below the liver there is all disease, and it disappears after treatment. So this study's going on, and we're hoping that we can improve our processing and our ability to generate these cells so that they're even more active. As I said, the CD7 is expressed in also AML, and this is just a graph showing that about 20% of patients express a lot of CD7 on their AML cells, and obviously many patients with T-cell non-Hodgkin's lymphoma express CD7 as well, so it's a good target for non-Hodgkin's lymphoma, which is even a bigger group of patients that have problems with this approach. And so we are opening a study targeting AML and T-cell non-Hodgkin's lymphoma with this same universal CAR T-cell product targeting CD7. And this has not opened, but it's going to open in the next month. And this just shows you that the expression of CD7 is not frequent. There's only 1 or 2 AMLs in this group that express CD7. That's on the far left on the upper panel there in this heat map. But when they do express CD7, they're killed efficiently with this product. Now, you might think, well, if you target CD7 and it's a gene that's expressed on NK cells and T cells, then what happens to all the T cells or the NK cells of the recipient? They'll all disappear, right? And what happens to the patients where they have no immune system? And so that is true. First of all, it's a little bit aspirational because CAR T cells don't persist forever. And so, but the reality is it will eliminate a lot of the NK cells and T cells, and it might do it for an extended period of time. So these patients could die from immune suppression. So it's a bridge to transplant. That's one approach. And also there's another way to get around that, and this was work done by Miriam Kim in our lab, recently published. And what Miriam decided to do, she said, okay, we know that mice, and that there are some humans born without CD7, and they seem to live a normal life. And it's a weird thing, but it's true. And there are some T cells in the peripheral blood that don't express CD7. We don't know what the function of those cells are. So what if we just knock out CD7 in the stem cell population and then do a transplant, and then transplant a mouse with CD7 negative stem cells? And so all the engrafted cells would come back and they would be CD7 negative, and they would be resistant to this treatment. The only cells that would remain sensitive to our CAR T would be residual leukemia cells and T cells in the patient. And so that's what she did. She took stem cells from a mouse and she knocked out CD7, and those T cells were engrafted. And what she showed in this experiment was that all of the B cells, myeloid cells, and T cells, and grafted completely normally, even though they didn't have CD7 and they didn't express CD7. And those mice looked completely normal and lived a normal life after transplantation. And then she showed in a separate experiment that when she compared the ability of this CAR T to target those engrafted mice. Now, there's two groups now, mice engrafted with wild-type stem cells that express CD7, and mice that are engrafted with CD7 knockout T cells, in which all the normal cells in the peripheral blood lack CD7. She showed if you compare the black lines there, those are the animals from the wild-type engrafted mice. There's a rapid reduction in all of the T cell numbers after treatment with the CAR T. And in the animals engrafted with the CD7 knockout stem cells, all the T cell numbers were present and they stayed the same. So they were completely resistant to targeting. So this is a way of getting her, and she showed also that when she did that, she could still target the malignant cells in these animals and kill them very easily. So this is a way to actually get around the long-term immune suppression that might occur in the context of targeting CD7. And this is being actually tested in the clinic now. So what if you lose CD7? So you could be doubting Thomas's, Debbie Downers, and say, okay, well, all these malignant cells, they modulate expression of a single target antigen. So we went after a second target because we knew this would happen. It would either be cleaved, downregulated, or spliced out, as what we've seen with CD19 in patients that relapse. And so we chose the second best target, CD2, and did the same thing. We knocked out the T cell receptor, we knocked out the target gene, in this case CD2, and we made a CAR to CD2, and so we made a UCAR-T2. And in the first set of experiments, I mentioned that we were studying a UCAR-T7. And this is very close to clinical development now, and we're about ready to go into the clinic with this. We showed that this UCAR-T2 kills CD2 positive T cells nicely in these in vitro experiments. But in vivo, something happened. In vivo, it turns out that CD2 in a T cell is very important for the T cell to function normally as an immune cell. It's a positive, it produces a positive co-stimulation or positive co-stimulatory signal in T cells. And you can see what we did in these experiments, we took CD19, which targets a B cell antigen called CD19, and we did the transplants with CAR T cells targeting CD19. And the only difference is that some of the CAR Ts had wild-type CD2, and the other CAR Ts had deleted CD2. We knocked it out. And if you look at the red line there, in vivo, the mice receiving knockout CD2 knockout T cells did not clear their tumors nearly as well. So this means that if you knock out CD2, you actually create a less effective CAR T. So we've got to fix that. Okay, so that's not going to be good enough. And so we figured out a couple of ways to get around that. I'll show you in a minute. So one was an observation made by two guys in the lab, Mike Rettig and Jerry Zhang. And they were looking at, we were trying to target CD2 and make our own antibodies, and they turned up these very old papers showing that there are some new antibodies generated to human CD2 that actually had a disparate binding to human versus baboon CD2. And so when we looked at the sequence of the human CD2 and the baboon CD2, we found that there were differences in only a couple amino acids. And so there were antibodies that were developed. We didn't even know about it. They were already developed that bound to the human CD2, but not the mouse, but not the baboon CD2, and vice versa. And so we took one of these antibodies that's being developed into the clinic now. It's called ciplizumab, and it's being developed for the treatment of people undergoing kidney transplantation. And it actually could recognize wild-type CD2, but didn't recognize the baboon CD2 that had those two mutations that were present at residue 18 and 55, so they are shown in red. And so what we did is we made our own mutations in those regions. We made a human CD2 that had the first mutation, and we made a human CD2 that had the second mutation and expressed these in NIH3T3 cells. And this is what we found. When we made these mutations, all the mutations were expressed normally on the surface, and so that was good. And then when we tested a combination of the single mutations or the double mutations, we found that when we looked at the double mutation, we had one antibody that couldn't recognize the double mutation. And when we checked another antibody, we found that that antibody could see that target just fine. So it's expressed on the surface normally, and it was visible to one antibody, invisible to the ciplizumab antibody. So this epitope is now what we call masked. It's silent. It's invisible to the antibody, but the antigen is there. So we've made a mutant that is invisible to one of the antibodies. And then we showed that this antibody, these two mutations together could produce a functional CD2 that actually bound to its ligand CD58 normally. And so we did this by flow cytometry. And we went on to show that the defect in the T cell function was completely resolved when we replaced the wild type CD2 with this mutant CD2, which had two mutations. And so now we're using CRISPR again in the context of adenovirus to replace the wild type CD2 with these double mutant CD2s. And now we have a CD2 which is invisible to one antibody but visible to the other antibody. So we can use this ciplizumab CAR T to kill all the residual host malignancies, but not the CAR T. So there's no fratricide. But also we can put it in the stem cells and transplant mice so that all the T cells that are generated have the mutant CD2 that functions normally, but is invisible to the CAR. And that way we can target residual leukemia without killing the normal T cell and NK cell population. So this is so-called epitope masking. And you'll see this, I think this is the beginning of this field moving forward. We're going to see a lot of this moving forward with CAR T cells, modulating the expression of certain antigens so they're invisible to certain CARs. All right, so I want to talk to you about one other way of enhancing CAR T cell function. And so there's been a lot of different kinds of ways to enhance the expansion, persistence, and efficacy of CAR Ts, and I won't go into these, but these are other people's work. We've been working on a ligand or a cytokine called interleukin-7, a long-acting interleukin-7, which, as you can see, is a fusion of several FC domains of two different antibodies to a genetically mutant IL-7, which makes it more active and also much longer half-life. The wild-type IL-7 has a half-life of minutes in the circulation, and this has a half-life of weeks. And so you can give it once every 2 to 4 weeks to patients, which we've done already. And it also enhances the expansion in vitro of CAR Ts, as shown by the upper right graph there. And we also show that you can do repetitive treatment of CAR Ts targeting a certain antigen in vitro. That's the bottom part of the graph. And you can see from the red lines that this allows for persistence of these CAR Ts for a longer period of time versus just unmanipulated CAR Ts. So these CAR Ts can last longer, can survive longer in vitro. And the question is, does this cytokine, is it worth its weight in gold in vivo? So this is the test. So we did the same CD2 expression, CD2 experiment, and we put in malignant cells that are expressing the target antigen CD2. That's the red line. And then the blue line, the solid blue line, is the animals that received CAR Ts to CD2. That's the universal CAR T2. And you can see that provided a substantial benefit to these animals, but they still all died from tumor. But when we added IL-7 only to that same amount of CAR Ts, you can see the dotted blue line on the bottom, none of these animals developed tumors, and there was no recurrence of the disease even out to 3 months after infusion. So we've done this with 2 other models, and it does the same thing. It profoundly impacts the expansion, persistence, and anti-tumor efficacy of CAR Ts. So what do you do when you get this information? You go into the clinic. And so we're in the clinic now, and we're in the middle of a multinational, multi-institutional study looking at the role of NTI-7, it's called long-acting IL-7, to enhance CAR T-cell efficacy in vivo. Because still 60% of patients or 70% of patients that get CAR T for large cell lymphoma relapse and die. So this is just an example of a couple of the early patients that have had complete responses to this added cytokine therapy. And it also increases the expansion of CAR Ts, as shown in the bottom right corner there, in vivo. The final limitation for CAR T-cell therapy, you've probably heard about it, is CRS, cytokine release syndrome. It is life-threatening and life-ending for some patients, and we have no idea what causes it. All we know is at the time that people are sick and die, they have enormously high levels of IL-6. Some drugs like tocilizumab can reduce that, but some patients even go on to die in spite of that. So we were interested in trying to develop a model to study CRS in vitro. And so this was really performed by 3 guys in the lab, Parmeshwar, Mike, and Rekha. And they did very simple experiments. They mixed CAR T cells, which were the effector cells, and the target population, which were, in this case, B cells expressing CD19, and the CARs express CD19. And so the target cells were bioluminescently labeled, so they're light, emitting light. And so if they die, the light goes away, so they're being killed. And then we added a cell called an IDC, which is simply a monocyte, because we didn't even know where the IL-6 was coming from. Most people thought the IL-6 that was being made in these patients is coming from a T cell, but we thought that there might be a myeloid cell involved. And so we then developed this assay and worked, and then we screened compounds that blocked IL-6 production, but did not block CAR T cell function. So that one, on this graph on the bottom right, it would be all those dots that are closest to the intersection of the Y and X axis. So that's the least CAR T cell impact and the most reduction in IL-6 production. And it turns out that 6 out of the 9 compounds were JAK kinase inhibitors or PI3 gamma delta inhibitors. And this just shows you 2 of them. This is divarasib, which is on the left side, I think it is. I can't see it from here, but it's divarasib on the left and ruxolitinib on the right. So PI3 kinase inhibitor, JAK kinase inhibitor, and they both inhibit IL-6 production, but have no impact on the bottom on CAR T cell function. And so we extended these studies to look at the role of the endodomain. Does it make a difference if the endodomain of the CAR is a CD28 or a 41BB, and it doesn't make any difference? And then we looked at what if you included these 2 drugs together. We found out that they're highly synergistic together. And so we then looked at, okay, so what's the biology here? We did single cell analyses and looked at and tried to figure out what cells were making IL-6 and what was going on here. So this is how it works. You know, when we did this experiment in a well, we put this IDC, which we thought might be making the IL-6, the CAR T, and the target population, and we did single cell analysis from that. You'd think it'd be a simple experiment, but they turned out to be multiple groups of transcriptionally distinct cells to look at. And you can see that there are 5 different subgroups of IDCs. That's on the top there. And then the T cells, there are at least 3 different big groups of T cells. And you can see that divarasib blocks the transition from that blue group to the red group. These are the activated T cells. So that is blocked. And also, in the case of the IDCs, the divarasib blocks the transition from one IDC group to the other. And it turns out that there's one IDC group called MDC1 on the top there. Those are the cells that make all the IL-6. And so all the IL-6 is made by one subgroup of IDCs, and those are the cells that cause life-threatening CRS in patients. And so the key would be to block that somehow. So either block the production of IL-6 from those cells or block the mechanism by which an activated T cell communicates with that cell to make IL-6. So the drugs divulisib and ruxolitinib block that pathway. And we tested this with bispecifics, and the same thing happens. So bispecifics, you know, if you've heard of blinatumomab, that's a bispecific. That's also a drug that actually induces IL-6 production. And you can see that this assay works in that setting. And it's the same inhibition of IL-6 production that you see with CAR-T on the left, you see with bispecifics on the right with these drugs. So it extends to bispecifics as well. And the final question you would ask, well, if you block IL-6 and in your little in vitro assay you're not showing any CAR T cell abnormalities, you're probably going to see that these CAR T cells don't work at all in vivo. So we did a number of mouse in vivo experiments and showed, at least with divulisib, that actually the CAR T cell function was improved, not decreased. So we think that this is an approach to take. And the final key here is that NK cells, which are another important effector cell that are being tested to treat AML, ALL, non-Hodgkin's lymphoma, and also to put CARs in them, that these cells don't produce any CRS. And one of the questions is, would this assay recapitulate that? And without a pointer, it's going to be hard for me to tell you, but you're going to have to believe me again, that NK cells in this assay don't result in the production of any IL-6 from these IDCs. So we think that there's biology here to understand that will be very interesting. So this is the first thing that you do when you see these things look good in vitro and in vivo. You do a clinical trial. And so this clinical trial is done now, and it shows that itacitinib, which is a JAK inhibitor, which we led, is an important inhibitor of CRS. And a second trial with divulisib is almost done as well, showing almost the same thing. The third trial would be to put these two drugs together to completely shut down CRS. And the final idea is, I told you that IL-6 is produced by these monocytes that have been made into these, what we call IDCs. They're monocyte, they're myeloid cells. They make all the IL-6. And so the question is, what tells them to make IL-6? An activated T cell tells the myeloid cell to make IL-6. So the question is, how does this happen? So we did a CRISPR screen, and we looked at all the things that might communicate from an activated T cell to a myeloid cell. And we found that one of the most important pathways was the CD40 ligand pathway. And when we tested this, we found that on the pink bars in the bottom there, you can see that if you block CD40 ligand, which is upregulated by activated T cells, that results in a significant drop in the production of IL-6 compared to the green line, which is the controls. And if you then test this in vitro, you can see that, and if you not only use an antibody, but if you actually knock it out in an activated CAR-T. So you take a UCAR-T and you knock out the CD40 ligand itself, knock out the gene, you can see that that results in a substantial reduction in the middle panel of IL-6 produced, but it doesn't affect CAR T cell function. So I think all of these, and this is just the same data showing that if you combine a CD40 ligand antibody with divulisib, you almost completely knock out IL-6 production. So that's it for universal CAR-Ts and targeting T-cell malignancies. And so I think the conclusions I would draw from this is that high-efficiency multiplex CRISPR-Cas9 gene editing of human primary T-cells and in human stem cells is feasible and safe. UCAR-T7 is effective at killing T-cell ALL, T-cell ALL cell lines in both in vitro and in vivo. And deletion or epitope masking of CAR T cells, or HSCs, allows efficient targeting of these diseases and without the concomitant worry of prolonged immune suppression. And long-acting cytokines, IL-7 in particular, but we're also looking at IL-15, may enhance expansion, persistence, and anti-tumor efficacy of CAR Ts, not just these, but all CAR Ts. And then mechanisms of CRS remain to be determined, as do optimal mitigation strategies. However, I think I've given you some ideas about how we might mitigate those mitigate CRS in the clinic as well. I want to thank the people that did the work, especially Mike, Jerry, Miriam, Julie Parmeshwar, Rekha, Julie O'Neill, and also my colleagues in the division who have been by my side from the beginning, and then collaborators at Woojin, Neoimmune Tech, Secura Bio, and my funding sources. Thank you very much. Happy to answer any questions.
Yeah. It was an excellent presentation, giving an insight for the CAR T cell with the perspectives of therapy. I want to know, like, what is the life of CAR T cell, and does the clinical trials show any recurrence after the CAR T cell life is over?
Yeah. So we know that CAR T cells can stick around for a long time. There was a recent paper by Carl June's group looking at some patients really several years after CAR T cell treatment where they could actually detect CAR Ts in them. But the answer to your question in reality is that they disappear relatively quickly, meaning that they're, at least in the peripheral blood, gone by 3 to 4 weeks after infusion. But if you do an animal experiment, they disappear as well. But then if you re-challenge the animal after that, especially if you give NTI7 or one of these things that enhances expansion and persistence of CAR Ts, then those animals are immune to re-inoculation with tumors, so that means that those cells are still in there. So that's really the more functional answer to your question. So that can be for a number of months. Yeah.
From the genomic perspectives, like there are mutations which are causing T cell. From that perspective, how does the CAR T cell, like once the patient is cured, but the mutation still is there, somatic mutation?
So I'm not sure, there's an echo in the room here. Did you ask me if there was genetic alterations in the T cells?
Yeah, genetic alterations, and if you're giving a CAR T cell therapy, like will it cure that?
Yeah.
I mean,
Yeah, so obviously there are genetic alterations that occur, and this has been of paramount interest from the very first studies done with retroviral vectors, which integrate preferentially into promoters, whereas lentiviral vectors do not. And so retroviruses can preferentially activate genes while lentiviruses don't have that significant possibility, but they still can cause problems. And I don't know if you know this, but in the thalassemia studies, there are several genetic studies done looking at lentiviruses to correct certain genetic diseases like thalassemia beta, and also some neurologic diseases in children, where children and adults have developed MDS and AML, and they found out that this was due to oncogenesis, insertional oncogenesis. So there is still a possibility that can occur. And so that's an issue. And then also for structural changes in the CAR Ts, we've done a lot of what's called ChromoSeq to look at big structural changes in the genes when we do the gene editing. And we really don't see frequent changes that are major, but there is always a possibility when you make 2 edits in particular that you're going to get rearrangements between those 2 genes. And so we look for that, but we haven't seen it. And so the FDA makes us look for these things when we actually want to do editing for 2 or more genes. The final answer to your question is that this could be markedly reduced by using what's called base editing or prime editing. Base editing, where there's only a single nick, there's not double-stranded breakage, as you see with CRISPR-Cas9, and so there's no breakage of the DNA and DNA repair. There's just a single-stranded break, which is annealed with great fidelity compared to CRISPR, in which the repairs are done very imprecisely. That's why you are able to knock out genes. So the movement of the field, I think, will be towards base editing. The problem with base editing is that it's much less efficient than gene editing right now.
Thank you.
Yep. Yes.
My question is, it looks like now you can engineer the CAR T cells, the universal CAR T cells for T cell malignancies. I don't know if I missed that, but then can you do this, the same thing for B-cell malignancies?
Yes, and they have been used for B-cell malignancies already. So there are gene-edited CAR Ts that are being used for B-cell malignancies. And the reason this was done was because when you have an acute leukemia, people are heavily pretreated, and all of the cells in the blood are malignant cells. And so capturing enough autologous T cells for gene editing or gene therapy is almost impossible. So the goal in that setting, even though it's a B-cell malignancy, would be to get T cells from a normal donor where they're healthy T cells as well. The problem there is that, they're not HLA identical, and there's going to be a graft versus host disease issue. So always from the very beginning, targeting B-cell malignancies was done just like this, by knocking out, at least in part, the T-cell receptor as well. So the T-cell receptor is gone, so those CAR Ts could not recognize the host as non-self. Yeah.
The question is, in terms of knocking out the T-cell receptor, can that be tried for garden variety allogeneic stem cell transplant to knock out the T-cell receptor, or is it because the donor T cells come up later on? Has that been thought of to reduce GVHD in allogeneic stem cell transplant?
Yeah, it can be done. It can be done, and the reason it hasn't been done is that we do allogeneic T cell we do allogeneic transplants now with reasonably good success. This is a lot of extra risk, obviously, to genetically modify a T cell population. But if you're asking me, if you knock out the T cell receptor, will you eliminate graft versus host disease as a potential side effect of the transplant? The answer is absolutely yes. Absolutely yes. The problem there is that would the result be that you would get more relapses because you don't have a graft versus leukemia effect? So that's a very important question. Problem is, to test that in a human would be phenomenally expensive, would take forever, and nobody has the juice to do that right now, or the money. But eventually, when we determine that editing the T cell receptor or deleting the T cell receptor is cost-effective and easy to do, then that would be a way to do it, and then you'd have to test whether the outcome at a year or 2 years, are there more people alive at the end of 2 years? Maybe there's a lot less graft versus host disease, but maybe everybody relapses, and so in the end, you might actually end up with a worse result, as you do with T-cell depleted transplants. So,
thanks.
One more question, I think. I'm glad there's a few questions. I thought there would be none, but that's okay.
So you had the phrase that we all hate, and then 6 months later they relapsed. Do you have any experience with repeat infusions, and is efficacy lost, and what's the escape mechanism?
I thought somebody would ask me that. Repeat infusions? Yeah, in the mouse we have. But remember, the mouse, we use an NSG model in the mouse. So the most of these mice are completely incompetent. They're immunodeficient mice, so they have no host immune system. So you can infuse CAR T cells a second time in the mice, and they're effective, unless the donor malignancy has lost the target, which can occur. It doesn't occur often in the mouse. It occurs 50% in human B-cell ALL and about 20% in human large cell lymphoma. So you lose the target. So that's a problem. That's why we went to CD2 as an alternative target, because we assume that this will happen, that malignancies will figure out a way to downregulate CD7 or lose it or shed it. And so we have to go after a second target because we have to have some backup. But yeah.
So have you established that that is the escape mechanism for?
I'm sorry.
Have you established whether that is the escape mechanism for the relapse then?
Oh, in the mice? So most of the, I was going to say in the mice, it's different than in humans. The loss of target antigen is much less frequent in the mouse. I don't know why, but just the way it is.
No, I'm a hematologist, so I'm interested in the humans. But you don't have experience with repeated infusions in humans yet?
No, so in the patients I can tell you one interesting caveat. This is very interesting. So you're asking me, in the humans, have we seen loss of target antigen? And the answer is yes. And we've seen one other thing that's even more interesting than loss of target antigen. So we've had a patient relapse. A patient had T-cell ALL. We put in UCAR T7. The patient had a beautiful response and then relapsed. And when the patient relapsed, the patient had, guess, for all of you, didn't have ALL anymore. Patient had AML. Because a lot of these diseases are stem cell diseases, and for whatever reason, they express, there are specific ones that they call them lineage infidelity or biphenotypic acute leukemias. So this guy was completely a CD7 positive T-cell ALL, and we eliminated those cells. But then when we looked at the cells were now expressing all these myeloid markers. And in retrospect, when he was diagnosed, there was a feeling that there was ectopic expression of myeloid antigens early on. So it was probably one of these biphenotypic acute leukemias or acute leukemias that are very primitive, that have ambiguous lineage fidelity. Yeah.
Okay, I think we'll leave it there. We're right on time, which is a sign of a real professional. So let's thank Dr. DiPersio again.
Okay.